Greetings, and welcome back to another episode of Purple Bear Biology. In this episode, we're going to talk about the scientific method. Have you ever stopped to think about what science is? Turn on the news and you're bound to hear some new research or what scientists have discovered. But what is science? How exactly did scientists perform the research? And how do I interpret it for myself? One of the first things I would like to start our conversation in this course with is the scientific method. While this may seem like a super simple concept that has been presented to you dozens of times, the scientific process is important to understand because it explains how and why scientists, including healthcare professionals, gather data or care for patients in a particular way.
Science represents the wealth of knowledge gathered through a series of steps called the scientific method. The scientific method consists of observations, questions, and predictions, and they're all a vital part of expanding our understanding of anatomy and physiology. Data gathering is also essential to document cause and effect relationships and provide support for conclusions. Collectively, conclusions can provide interpretations of trends and share that knowledge with the rest of the world. The whole process seems so exciting. So let's get started talking about the scientific method. Now, I know it has probably been a while since many of you have thought about the scientific method, or maybe this is even the first time you've explored the concept. Either way, you should know that you use the scientific method every day as you problem solve to find solutions. The steps in the scientific method include observations, a hypothesis or a prediction, an experiment to test that prediction, gathering results to determine whether or not you support your hypothesis, and then, if you support your hypothesis, you perform the experiment again to increase sample size and test your results, or if your results do not match your predictions, then you revise your predictions, creating a new hypothesis, and perform either a new experiment or the same experiment over to test to see whether or not your results will now match your hypothesis. While these are the general steps that you can find in the scientific method, they all kind of seem detached from our everyday lives. So let's look at an example that you all know and may have encountered once in your life. Let's use the example of if you walk into your bathroom and you flip on the light switch and the light doesn't turn on. Your first observation would be that you're in a dark room, and when you flip on the switch, there is no light. You then would proceed to making a prediction. The power is out. Maybe the power is out to the entire house. To check this, what would you do? Well, you would design an experiment and check the other power sources. Now, you wouldn't think of this as an experiment. You would just go around and start flipping light switches in other rooms to see whether or not the lights came on. Maybe you would check the digital clock that you use as an alarm clock that's by your bed. Or you'd walk to another room and see whether or not the fans and light sources work. All of this is experimentation testing whether or not the power is out to the entire house. So essentially what you're doing is gathering data to see whether or not it supports your prediction. You would then use all of this to draw a conclusion about whether or not your house has power. Let's imagine that you get all the way to the end and you find that all of the other lights in the house turn on. Well, then that would not match your prediction that the power was out to the entire house. So, what you would do is revise your prediction and come up with a new explanation for why the lights won't turn on in the bathroom. One example might be that maybe it's just the bulbs inside of the bathroom that don't work. And so then you would have to perform a new experiment to test this particular prediction. You could replace the bulbs inside of the bathroom. You would go through multiple processes or steps in order to see whether or not you could determine what would be the overall effect causing the lights not to work. In all of these examples, what you're essentially doing is making observations and then predictions based on those observations with the information that you have available to you. This is why oftentimes a hypothesis is referred to as an educated guess, because it uses the information you have available to make a prediction based on the observations that you had made. All of this is the scientific method, and you use this to problem solve tons of things throughout your everyday life.
Okay, to further explore this concept of the scientific method and kind of solidify it in an example, I brought a vase in here with me today. Okay, so now this vase, it has nothing on top of it, nothing on bottom, right? There's nothing spectacular. I don't want to turn it over too much, but you can kind of see there's nothing on bottom. There is water inside of the vase. And so it's almost full, you can kind of see. What I want to do is test your ability to use the scientific method. So let's start with this glass here, and I'm going to pour some water from this vase. I'm going to pour it all out, right? I'm going to pour it all into there till there's no water left. Okay, a little bit of trickle there. Okay, so we can see no water left, right? Now, that filled it up, right? That was kind of neat. Now, oh yeah, wait, there's more water in there. Okay, there's definitely all the water's gone now, right? Oh no. Wait, there's more water in there. Well, let's, I got another glass. Here we go. All right, so let's empty all that out. Every last drop, right? I mean, that was a lot. Now, of course, oh my goodness. Well, there's more water in there. Now, one of the interesting things about this little experiment is that every time we clearly are making an observation that the water is all gone. So let's, oh my goodness, what in the world is going on here? There always seems to be more water left in the vase. Now, if we were to use the scientific method for this, right? We're making an observation. We saw there was water in the vase. There's nothing around here, right? We saw that all the water seems to be coming out of the vase, right? It's completely empty, but then whenever it's not completely empty, right? I don't understand that. It seems so strange. Now, I'm sure some of y'all are making predictions about this. I'm sure some of y'all are like, okay, well, there's got to be something going on here, right? One possible solution is that it's magic. One of the first things that you might do, of course, is then start examining the vase. Right? You'd be like, okay, well, I want to look at it now. Of course, you're in a digital space, so you don't get to do that, right? But if I were to take this vase and allow you to examine it, you might notice that on one side, there's a little hole here, right? Now, what's really cool about that is that if I put my finger over that hole, it actually creates a little bit of a vacuum inside of the container. So if I then take and I pour this out, the fluid that is inside of there, that is not directly inside of the column, is inside of a vacuum. Now, if I let that go and tilt the cup backwards, it relieves the vacuum, and then out comes more water. Isn't that amazing? Over and over and over again. So there was something about the system that we didn't understand. In science, oftentimes this is what's occurring. We have things that we can't directly observe or haven't figured out yet, but we can see an observation and we want to try to understand how it operates. So we make these hypotheses or predictions, we design experiments, and then we perform those experiments, gather data, right? And then if it doesn't match our prediction, then we revise that hypothesis and do it again. If it does match our prediction, then we try it all over again just to make sure that what we came up with wasn't some natural variation that just randomly supported what we said. And then you perform it again and again and again. Now we're going to continue to talk about this concept so that we can understand the scientific method and how it develops into theories. So let's jump right back into the slides and talk a little more about it.
First and foremost, experiments need a control group. This allows you to determine if the variable of interest is having an effect. The purpose of the control is to create a standard to compare experimental results to. Let's use an example to understand the importance of a control. Imagine that you make a bad decision and you eat some gas station sushi on a quick lunch break. After a few hours at work, you begin to feel nauseous, so you go home. You spend the next 24 hours just vomiting and hugging the toilet. You can conclude that the sushi caused the illness, right?
Well, not so fast. Without a control, you do not know if it was the sushi or something else that you encountered that day. Maybe a coworker had the stomach bug and you contracted it from them. Without something to compare your situation to, you cannot conclude it was the sushi. If you checked with your other coworkers that did not eat the sushi and they were not sick, then this would be a stronger position that the sushi was the cause. Or if you found other individuals that ate the sushi that did not become ill, then you could begin to conclude that the nausea was caused by something else entirely, something from the environment. Controls are incredibly important when determining the effectiveness of treatments for illness. We must have a control group with individuals that receive no treatment and another that receives treatment so that we can compare and determine the effectiveness of the treatment overall. In addition to control groups, we have to have the ability to quantify a treatment's effect. When we consider quantification, we have to consider sample size. Let's venture into Microsoft Excel and explore sample size for a minute.
All right, so now that we're in Assign in Excel, let's talk about sample size. The sample size is the number of individuals you have in your study for each group. For example, if you had 10 individuals in a study, half were in the control and half were inside of the treatment group, then you would have a sample size of 5. 5 control samples and 5 treatment samples. Sample size can have a huge effect on perspective of effectiveness of treatment. Let's use this example to explain what I mean. Imagine you want to test the effectiveness of a new drug that helps regulate blood pressure. Small sample sizes tend to show variation dynamically. Let's take a look at the systolic pressures of a sample of 5 individuals. First, notice that when we look at an effect, we graph averages, not the individual points. Individual points may be skewed by unknown variables. Averages with adequate sample sizes tend to be more representative of a population as a whole. You can see that on this data, there's little difference between the averages of these samples. Adding extreme values, though, can quickly disrupt this trend. Imagine that we added a few more individuals that had 158 as their systolic pressure. Notice that those individual values start to increase the drug's average overall. While this may seem like a bad thing, increasing sample size can actually help balance out extreme values and give us a better picture of the actual effects of our treatment. To demonstrate this, let's add some more samples to this overall treatment. Let's add another 10 individuals to our treatment, and now let's take a look at our averages. Notice that the averages for these are now very dynamically different than what we originally started with when we had a sample size of only 5 individuals per treatment. In this case, it now appears that the drug is having a positive effect at reducing blood pressure, whereas there didn't appear to be much of a difference before. Why is that? Well, it has a lot to do with the fact that before we added in these additional 10 treatments, within those 5 samples, we had a really high value for drug A. And because our sample size was so low, it caused the entire average to be elevated. But when we add additional samples in there that are more representative of all of the individuals that may be taking the drug, we can see that there's actually an effect that causes the drug to reduce blood pressure rather than there being no effect at all. So you can kind of get a perspective on how sample size is so vitally important when we design experiments. More samples means a better representation of the population as a whole, and extreme values won't have as significant of an effect on our overall averages that we take a look at.
Okay. So now that we have discussed the scientific method and some of the important details to pay attention to, let's finish this video by discussing what happens when your experiment supports your prediction. The short answer is you do it again to see if you get the same result. But if you confirm your results multiple times, then what?
Well, you share that with other scientists and they test your predictions. And the process repeats itself. And if no opposing predictions are presented by the scientific community, then the thoroughly tested prediction becomes a theory. It's important to understand that theories in science represent something that has yet to be disproven, but it has been tested by numerous individuals multiple times. Theories from this perspective are incredibly powerful predictions.
Does this mean that theories cannot be proven wrong? Nope. It only takes one valid result to disprove a theory. Only one! It is this adaptability that allows science to progress forward as our understanding of our world develops. So let's summarize what we have explored in this video. First, we use the scientific method to explore everyday problems, but it is a powerful tool in determining the effect of treatment and has a huge influence on how medicine gets practiced. Second, Experiments are considered stronger when they have controls to compare their results to. When a sample size is large enough, it tends to account for the natural variability that we have in a population. And third, theories are examples of rigorously tested predictions that have yet to be disproven. Well, that's it for this episode of Purple Bear Biology. I hope you learned something along the way. If you enjoyed the video, be sure to hit the like button below and drop a comment in the comment section, and subscribe if you want to follow to see more videos that I release about anatomy, biology, and microbiology.